The overall goal of this procedure is to obtain native oral biofilm from palatal expanders for fluorescence in situ hybridization. This is accomplished by first removing the palatal expander carrying the 4-month-old biofilm from the patient. Next, the acrylic resin flakes containing the biofilm are scraped off from the palatal expander. Then, fluorescence in situ hybridization of the fixed biofilm is carried out directly on coated slides. Finally, analysis of the stained biofilm is performed using Confocal Laser Scanning Microscopy. In the end, the structure and composition of bacterial biofilm communities are shown through fluorescence in situ hybridization and confocal laser scanning microscopy.
We first had the idea for this method when we encountered thick biofilms on orthodontic expanders visible to the naked eye. They made us think of possible associations between them and periodontal diseases.
This method can help answer key questions in the field of oral microbiology, such as the spatial arrangement and composition of native oral biofilms. The method is interdisciplinary and has to be done by different specialists, so Elizabeth is an orthodontist
and Barbara is a molecular biologist.
To begin, after 4 months of intraoral surface, using the Nola dry-filled system, isolate the fixed palatal expander. To avoid contamination, remove the appliance from the patient's mouth using sterilized pliers, gloves, and trays. Store the objects in 120 ml vials at -20 degrees Celsius. Take them to the laboratory on ice and process within 24 hours. To fix the specimens, scrape off biofilm flakes with a sterile scalpel or collect pieces with sterile forceps. It is crucial to scrape off acrylic resin flakes of adequate thickness to ensure that the biofilm structure remains intact. Add ice-cold 4% paraformaldehyde solution until the sample is well covered. Incubate the mixture at 4 degrees Celsius for 3 to 12 hours. Longer fixation times or higher fixation temperatures may render the cell envelopes of gram-negative cells less permeable to oligonucleotide probes. After the incubation, remove the paraformaldehyde solution and wash with ice-cold 1x PBS. Repeat this step 2 to 3 times to remove residual paraformaldehyde. Resuspend the sample in 1 volume of ice-cold 1x PBS and add 1 volume of ice-cold 96% ethanol. Store the sample at -20 degrees Celsius. Samples can be stored for several months to years. To dehydrate the sample, first apply 5 to 30 microliters of paraformaldehyde fixed sample material to a microscope slide. Dry the sample for 15 minutes at 46 degrees Celsius or for longer at room temperature. While the sample is drying, thaw the lysozyme and the formamide. To partially break down the cell walls for better entry of the probes, add 250 microliters of a 1 mg per milliliter solution of lysozyme for 10 minutes at room temperature. Dip the slides into 50, 80 and 96% ethanol for 3 minutes each. The dehydrating effect of the ethanol concentration series will disintegrate the membranes for better oligonucleotide probe uptake. Finally, dry the slides at 46 degrees Celsius. To perform in situ hybridization, start by preparing 1 ml of fresh hybridization buffer and pre-warm it to 47 degrees Celsius. Thaw oligonucleotide probe solutions on ice and keep them protected from light. Place a piece of tissue paper into a square petri dish and pipette 800 microliters of the hybridization buffer onto the tissue paper. Close the chamber again immediately as the buffer contains formamide. Add 2 microliters of each probe to 200 microliters of the rest of the hybridization buffer. Mix well and apply the mixture to the dehydrated sample on a microscope slide. Immediately, place the slide horizontally into the dish and cover it. Incubate the dish in an oven for 1 to 5 hours at 46 degrees Celsius. The dish functions as a moisture chamber, preventing evaporation of hybridization solution from the slide. In particular, the evaporation of formamide can cause non-specific probe binding to non-target cells. Prepare 50 ml of washing buffer and preheat it to 48 degrees Celsius in a water bath. Remove the dish with the slide from the hybridization oven. Immediately wash away the hybridization buffer with a small volume of pre-warmed washing buffer and transfer the slide into the remaining washing buffer. Place the tube containing the washing buffer and the slide back into the water bath and incubate for 10 to 15 minutes at 48 degrees Celsius. To stain the samples with more than one probe, repeat the earlier steps after the washing buffer has been removed. Take care to start with the highest formamide concentration. Remove the slide from the tube and dip into ice-cold double distilled water for 2 to 3 seconds to eliminate residual washing buffer. Air dry the slide as quickly as possible, as fast drying reduces probe dissociation. Store the dried slides in the dark at -20 degrees Celsius for several weeks without any significant loss of probe-conferred fluorescence signal. To image the samples, apply 2 drops of anti-fade reagent close to the left and right ends of a slide and then add a cover slip. Observe the samples under a confocal laser scanning microscope equipped with suitable filters or lasers. The slides can be stored at 4 degrees Celsius for up to 7 days or can be washed with double distilled water and stored at -20 degrees Celsius for longer periods. Scraping biofilm off fixed orthodontic appliances yields suitable flakes that can be hybridized directly on coated glass slides for microscopy. Such different groups of oral microbiome bacteria in 3-dimensional configuration can be identified through specific probes. In this figure, the biofilm was stained with EUB mix and LGC mix. Firmicutes appear in green, as they were stained with yellow and blue. Here, the biofilm was stained with EUB mix, BAC303 and POGI. Bacteria of the group Bacteroidetes appear in pink, and Porphyromonas gingivalis is shown in yellowish white, as all 3 probes bind to its DNA and the overlap of colours results in a white signal. Morphological differences between groups of bacteria can also be identified, as well as certain structures like large clusters of coccoid bacteria. Examples of such clusters are shown here, where EUB mix was used for staining. In addition, a typical mushroom-like structure of the biofilm can be processed via 3D modelling of the CLSM data. This is a 3D animation of the EUB mix stained biofilm.
This method can provide insight into orthodontically induced biofilm formation. It can also be applied to any other artificial surface in the human body that carries complex biofilm structures with possible pathogenicity.
After watching this video, you should have a good understanding of how to prepare native oral biofilms for fluorescence in situ hybridization and confocal laser scanning microscopy. It's important to remember that this is an interdisciplinary field, and you can only be successful in a team, so always treat your colleagues nicely.
Take into consideration. Sampling and hybridization are the most critical steps. During sampling, Care must be taken that slices of adequate thickness are collected to ensure that the biofilm structure remains intact. During hybridization, it is essential to avoid excessive fluctuations in temperature, thus avoiding nonspecific binding, or loss of binding, of the fluorescently labeled probes. This FISH protocol is an elegant method to stain normal heterogeneous biofilm directly on glass slides without disrupting its composition. The slides can immediately be used for microscopy without a need to move the sample once again. Thank you for watching. Subscribe if it is useful.